Bismillahirrahmanirrahim. In this video, synthesis of hormone, especially the polypeptide and protein hormone, is discussed. Most of the hormone in the body are polypeptide or protein hormone. They are stored as secretory vesicles until they are needed. These can be divided into proteins, or generally it can be divided into protein or polypeptides. If they are comprising of more than 100 amino acids, they are termed as protein, or if they are less than 100 amino acids, they are termed as polypeptide hormone. So examples of these are thyrotropin releasing hormone that contains only 3 of the amino acid, while as many as 200 amino acids can be present, that is seen in growth hormone and prolactin. These hormones are synthesized in the rough endoplasmic reticulum as pre-pro hormones that are biologically inactive. Then they are converted into prohormone by enzymatic processes. They from the rough endoplasmic reticulum, they go to Golgi apparatus, and here they are grouped as or packed as secretory vesicles. These in these secretory vesicles, they are composed of, or they contain, active hormone as well as inactive fragment of the hormone. And in these secretory vesicles, they are present within the cytoplasm under the cell membrane. Whenever there is increased intracytoplasmic calcium concentration, that will result in exocytosis, or whenever there is increased cyclic MP, that will result in increased activity of protein kinases. It will result in exocytosis of these secretory vesicles, and the hormone, along with the inactive fragment, comes into the interstitial fluid, and from there it goes to bloodstream, and circulatory system now carries this these hormone to the target tissue to produce the effect. So revise this synthesis of these polypeptide hormone. They are synthesized as pre-prohormone in the rough endoplasmic reticulum, and after the cleavage into the endoplasmic reticulum, they form prohormone. These prohormone are transported into Golgi apparatus and packaged into secretory vesicle, and then the final cleavage occurs that contains active hormone plus inactive fragment within the secretory vesicle, and this is done by this Golgi apparatus. And these hormones are stored in the vesicles at the cell membrane cytoplasm. The trigger for their release is either calcium influx. Membrane depolarization leads to increased cytosolic calcium that triggers exocytosis, or receptor stimulation by ligand that binds with receptor, and then cyclic MP is increased. That results in increased protein kinase activity, and that will result in exocytosis. This image is diagrammatic presentation of biosynthesis, processing, storage and secretion of polypeptide and protein hormone by endocrine glands. It captures the intracellular steps that are involved from gene transcription to the hormone exocytosis, specifically for peptide or protein hormones. So how the synthesis starts: from the DNA, messenger RNA is formed by the process of transcription in the nucleus. The gene coding for the hormone is transcribed into the messenger RNA. So the messenger RNA will be will be responsible for the translation process, and the messenger RNA exits the nucleus and binds to the ribosome on the rough endoplasmic reticulum, where translation produces a pre-pro hormone. So this pre-pro hormone are then cleaved into prohormones, and then the prohormones are packaged in the Golgi. They are first transported and then packaged into the Golgi apparatus, where they are further processed, and they are now pegged into secretory vesicles. Here these purple dots are secretory vesicles. So these secretory vesicles are enzymes that have got cleaved prohormone that uh, contains active hormone along with inactive fragments, and then uh, these are stored within the cell, often just under the plasma membrane and ready to release upon stimulation. So when there is stimulus for their release, there are two major stimuli or two major signaling mechanism. One is increased intracellular calcium concentration or increased cyclic AMP concentration. These signal triggers the fusion of vesicle that are lying into the cytoplasm or just beneath the plasma membrane. They start the fusion of vesicles with the plasma membrane, lead to exocytosis of these vesicles into the extracellular fluid, where it enters the bloodstream. This is the process that is regulated. It is not constituted. It is regulated, and this hormone secretion occurs on demand. It happens on demand. Peptide hormones are water soluble, allowing easy circulation in the blood, and this inactive fragment that is co-secreted with the hormone, it has got a diagnostic importance because it can be used for diagnostic hormonal assays. Thank you for watching this video. If you have any query, please write in the comment box.